we have the honor to introduce an scholar, researcher, Dr. Chandraiah, who serves as an assistant professor, National Institute of Pharmaceutical Education, Research, in the Department of Biology, specializing in regulatory toxicology. Dr. Godugu holds an in pharmacology and toxicology from Mohali, a PhD in the same Mohali, awarded in 2011. He holds a postdoctoral fellowship at Florida A&M USA in the year 2013. He has over 10 years of research and teaching experience and is a diplomate of American Board of Toxicology, ABT. His multidisciplinary research spans pharmacology, toxicology, drug delivery, nanotoxicology, with a special focus on respiratory disease. Chandraiah has been in Stanford University's top 2 scientists in pharmacy for 4 consecutive years, that is from 2022 to 2023. He authored 200 publications and contributed to book chapters with over 7,400 citations, a high head index of 44. He has also been the recipient of multiple awards, including the prestigious Dr. Mukherjee Award for the best paper in the Indian Journal of Pharmacology, the DP Patil National Best Thesis Award in 2020. Dr. Chandraiah has successfully guided 60 master's dissertation students and 20 PhD students. Dr. Chandraiah is a distinguished contributor in the scientific community, renowned significant contribution across various fields of research, whose expertise spans various disciplines and collaborative efforts. He has successfully secured extramural fundings for 13 significant research projects, including grants from prestigious organizations like the Indian Council of Medical Research, ICMR, Department of Biotechnology, Department of Art, and international collaboration as the German Joint Research Grant, CDAD, the DBT Indo-Brazil Joint Project, the DBT Northeast Twinning, and the Science Engineering Report, SEB. His work as principal investigator has attracted over 5 in research and as a principal investigator on crew. In his impressive funding achievements, he holds multiple patents and is very recognized for his expertise in criteria, drug discovery, organ analysis, cancer development, toxicology of nanoparticles, distribution, and well academia, making him a prominent personality in the community. We are incredibly fortunate to have him with us today. We extend a warm welcome to the speaker session, Dr. Chandraiah.
Thank you, Harita ma'am, for that nice introduction. Sir, I request Dr. Chandraiah session.
So, at the let me a wonderful introduction. Actually, she worked on resume. Looks like See, some of the words using not making my resume. So, thank you so much for very surprising introduction about me. To all the participants joining from different farm places across India. I can see 3 participants joining, and I was told that 300 people have joined. Good morning to all. I know you all be contributing to the field, wherever you're working, and looking forward to interact with one hour about some of the upcoming things in our farm field, integrate ML into drug discovery. So before actually going into my presentation, let me thank the organizer, Dr. Malik sir, principal of the college, Dr. Muradu, whom I met months back in one of the event in high-tech city. And Samyukta, she called me over a phone. She was asking, requesting for the talks. It could materialize in a or 2 months, and very happy to be part of this particular the faculty development program on artificial intelligence in public health. How we are going to improve the curriculum of the profession. This is really a very good topic you have chosen, and let me congratulate, let me appreciate the efforts of the MLR guys. Select a very apt title and a theme of the fact development program. More students in the family are actually in some area. I think they can really we don't have the proper high-end laboratory setup also can actually do good research work just based on artificial intelligence and machine learning. Hope I have things. So before actually before that, let me share presentation. That I think we can we will go ahead. Uh, so shall I share my screen?
Yes, sir. You can share.
Okay, time to share. Okay, some difficulties like. Can you able to see screen?
Yes, sir. It is visible.
Is it moving?
Yes, sir.
Is it moving?
Yes, sir. Everything is perfect.
Perfect. No, if you're not able to hear me, please stop there. It can be a two-sided interaction. I'll be very happy. I know you're not students, you are the faculty. I don't want to be all the minor things. Only want some direction. Now we can do differently than what I've been doing in pharmacy profession, right? Title of my talk is opportunities in drug discovery and development, era of AI ML, right? Artificial intelligence, machine learning. Trust me, spending almost 2 years in why I'm saying I joined in Mohal in 2004 and today is 2024. So 20 years of my journey into networks, including years of postdoctorate. Uh, there was no great period in the current period what we have today. The period is of more of a artificial intelligence and machine learning where we can actually uh, reduce a lot of time as well as a lot of experimental work by using these tools. Right? I feel go up the drug discovery development, if at all, if you can think of, right? And many people, many foreign universities or many companies are already into this area and trying to get a benefit from this particular artificial intelligence and machine learning. So with this the things happening across the world or in India, why we should not think of, if you talk about any software company, I visited a Capgemini company 2 months back, and there was a panel on role of AML in drug discovery and development. See, I have not significantly connected to the AML, but they have invited one of the panel to discuss how we can take it forward, and it's also really trying to establish this AML centers in a big way. If the center comes, the prime uh, area should be occupying pharmaceutical science. So we all on this. That's why I also tried to kept the image, which is a with the theme, right? They have a tablet. We also have a lot of chips and the computer language, right? That indicates that to integrate a computer, the tools or software into our drug discovery or development. That's what actually the is going to happen in the next at least few years. Maybe 5 years we'll have a lot of to happen. It. By the way, let me also tell about this my qualification in the DBT. DBT is a diploma in American Board of Toxicology. which is very tough exam to clear. I'm very proud to say that being a toxicologist, I have this exam and I wrote this exam and I have cleared. I'm a certified toxicologist. I can actually review the toxicology report generated by different pharma companies. Right. So that's what my background is. So not without wasting much now, let us actually jump in because I have more than 90 slides. Some of the slides probably we can skip it if they're not relevant. Um, we all know it. What is that? So I hope everything is clear, people, right? So just want to have a confirmation. All visible and we're on the same page.
Yes,
All
sir.
good?
Yes, sir. Everything is
Great.
going
Okay.
good.
So please, I cannot see any that's why you should stop me, right? Something is not good, you should stop me, right? So on drug discovery at a glance, we know that it takes more maximum it can be 15 years and it's costing 10 US dollars. That is equal to more than 50 crores of money in rupees, and it's a high risky effort. That's why no in my company was to venture into this. I wanted to have a safe game. Okay, I will have a lab. I'll just do some a formula which is already existing. I'll do generic thing. I'll do some abbreviated new drug formulation and or I'll just only make a very incremental work. You don't want to do the very hard the drug discovery development because it's very costly, very risky, as well as if it goes wrong at late stage, company will not able to survive. That's what happened to some of the major pharma companies, including Dr. Reddy's. There was a setback when Valley developed a anti-diabetic drug, balaglatazone, that got failed in the phase 3 trial, phase 2 or phase 3 trial, and they have to really change all the the policies later. Then they focus more on something else later. So what I'm saying is it's very risky business. That doesn't mean that we should think of the drug discovery development. Only Pfizer's, only Sanofi's, Novartis will do the drug discovery, and Indians only appreciate the efforts and we just contribute to them. If Indian pharma graduate working in India in multinational pharma companies, they're all working for them just as a, giving a lot of service to them, but we are not actually contributing to Indian growth in the form of drug discovery and development. Just tell me how many new drugs have come from India. Why drugs cannot come? Is it a lack of the manpower, lack of the what is lacking? We have such a wonderful pharmaceutical industry. Still, we are not thinking of drug discovery because these limitations, AML, and you all know that can come from any source, or you can also do the repurposing. And these steps, I don't want to talk about it because for last 4 lectures, I'm sure somebody might have talked about it. All might have talked about it. Me talking about it is not going to make any sense. And also going to see this as well, drug discovery and development. What is a discovery? Lead optimization, you can call the discovery where target selection, target validation from the lead optimization, it takes 2 to 6 years. So this is clinical stage basically, more of a discovery stage, 2 to 6 years. So this 2 to 6 years can you do less than one by using a tools like AI. Is it possible? It was not possible earlier. But now today it's possible. See the numbers: 10,000 to 20,000 molecules are required to get one drug. Are you lucky drug approved? So 10,000 molecules, who is going to synthesize it? How are you going to characterize them? How are you going to screen them? Are the tool? These are the challenges we have been facing earlier when we are talking about the drug development. So we able to aid yes, a company to spend a lot of money, just keep on same paying salaries, keep on paying a lot of consumable cost, keep on instruments. I think needs to have to maintain the animals, we have to maintain the GLP, good laboratory practices. Imagine what kind of in that. That's why actually the right only if they only doing investing the money, no rate, how long they will do? Can we able without us for 1 year? I mean, I'm talking about the family. But imagine in a company having employees for years, uh, they're not sure they're going to get it approved or not after 10 years. What is going to happen? That's why be walking away. That was the scenario earlier. But today, things have changed. Discoveries can happen at the academic level. Discoveries can happen at institutes like your organization. The conceptualization can be happening at the institution Malared College of Pharmacy in integration with the engineering background people. So electronics background people, medical device background people. I think it's possible today. And how it is possible? Using the current tools like AML tools, right? So then discovery is 2 to 6 years. We can reduce it, and also developmental phase. What is the developmental phase? We're going to do the extensive preclinical tox studies to see that the molecule is safe or not. If the molecule is not safe, you cannot take that molecule further to the clinical trial. So, the phase one, phase the trial, how it is happening, and how the NDA approach are also taking place. So, target selection, are going to use it, lead discovery, how are you going to do the synthesis, how are you going to do the combinatorial chemistry, how are you going to do the assay, and also can we have the high throughput, talked about traditional chemistry, in vitro screening is been practiced, in vitro studies, drug affinity, how it is binding to the target, is it selective to the target or not, will be done. Then in the animals for pharmacology as the toxicologist was done, then clinical trials will be done later. So do are there is going to be a lot of dash taken at the lead level, at the target level. Every time you select a target, every time you do the screening on a molecule, then there has to be a meeting happening saying that whether should we go ahead or should we go back. Because it's very dangerous to take a molecule which is having a problem. If you are not discussed early, the molecule will be traveling another 1 year. Imagine you have missed some of the important aspects which may be a problem later, and you are not thoroughly evaluated at the early stage of the uh, science, and that molecule uh, has been given a clearance to evaluate for another phase. It can be animal detailed animal toxicology studies. That 1 year or 2 years of your time may be waste, right? Because the molecule is bound to fail later because you have not evaluated. That's why you need to have a strong team of the chemical, biology people, toxicology people, population people, whatnot. Every regulatory, all have to. They need to also see the regulatory aspect, right? Because you cannot take a drug which is new drug. We are spending this of time. If you know, not just, if you are able to bring a uniqueness in that new drug, what is going to take a molecule? That's why you need to have multidisciplinary team working in organization doing it. And that's why so early killing is very important. If you're able to kill the molecules early, just killing. If you have to kill it early. If you don't want to kill it, make sure it's not going to be killed later because of the issues. Why drug will be killed in the sense, withdrawing from the further developmental phase. Why we will be withdrawing from the development phase? It may be not feasible to use for human applications because of the formulation not suitable. It's not viable because not take injection every day. It may be having certain interaction in the body, producing toxic effect in the humans later, which is not to be uh, identified in the animal trial. So whatever it may be, so we have a lot of things to be done, and the go or not, this decision needs to be taken. Whether you need to be very sure whether you want to go to phase one trial or not, because the moment you enter the clinical trial, 70 to 80% of your cost is going to be put in the for the clinical trial. That's why has take compared to the discovery phase, right? 2 to 6 years maximum period, but now a clinical trial period will be more than 8 years. So that lot of money is required. Why a lot of money is required? You don't do a lot of science in the clinical trial. But why you spend a lot of money? Because directly patients are involved in this, and you evaluate all the profiles of drug exposure, how long you need to monitor them, and how the patients and the clinical trial participants are adhering to the particular study protocol. Because it's not just you take a drug and you'll walk away, right? You need to monitor for the next the period to any unwanted issues happening because of the drug exposure. So these all things will result in the highly increased cost. Cost can R&D cost are increasing. the preclinical cost as well as the clinical cost. Both will be increasing it. Why both costs are increasing? Because to talk about the preclinical costs are increasing. The reason for increasing preclinical cost is earlier there was no GLP required. Earlier there were no the number of toxicology studies needs to be earlier maybe 1998 or 2000 per lesser compared to today. Now, if you say the ICS safety guidelines, ICS efficacy guidelines, just imagine how many number of up to 11 guidelines. Each guideline will have battery of the toxic study to be done, battery of the toxic studies to be done. That means no, the regulator is not going to consider your molecule clinical trial until unless it all the required battery of the toxic studies, carcinogen studies, local toxic studies, skin sensitization, reproductive toxic studies, toxicokinetics, safety pharmacology studies, PTO prolongation, cardiotoxicity, whatnot. We have so many number of toxic studies done. So the number of toxic studies required are only going to increase, not never decrease. Now, so that's where a lot of your R&D cost is going. Time is also increasing it as the cost is also increasing it. And the clinical trial cost is increasing now because earlier were not highly no, in the sense not highly no, costly at first. But today it's very costly because if one country is done the clinical trial based on the data, you can also get an approval in other country. But this is not the case today, right? You have to do in the each geographical location, in each country you have to do it. Now the type of population also increased it. Number of patients to be involved in also increasing it. And you may compare with the comparator drug. Earlier there was no drug available, right? So there was no need to compare it. Now you need to compare the comparator drug, existing drug. What kind of efficacy, what kind of safety your drug is going to offer. So this all resulted in what is hard time time. So same thing. It's going to cost more than $1 billion, 14 years. That's why AML is transforming the discovery pipeline by enhancing the efficiency, reduce the cost, and accelerating the. So that means it is going to reduce cost. It is going to reduce the. It is going to increase the efficiency of that discovery. It's not that 20 molecule, 1%, one molecule getting hit. 100 molecule, maybe maximum 500 molecule, maybe you can get drug approved without any issues. And also timelines are going much short, not 14. It can be a couple of years. Of course, you cannot reduce the clinical trial duration, but there are ways you can actually design the clinical trial in such a way that from 8 years, probably you can use to 4 years. Or you can get the data from the existing literature saying that, so based on parameters, I think that no, my trial is safer. You are going to use the AML tools in the clinical trial because each person is going to produce huge amount of the data, and now thousands of people are going to participate in clinical trial. How the human leads are handled, how the data can be handled, that's why you have to use the AML tools. So these are all going to use increase the efficiency of the drug discovery program. What are the different areas AML has been used in many aspects of drug discovery and development? At the drug design level, you can use it. Drug design means what do you mean by drug design? No, you are not even synthesizing it. You are just designing by using the in silico tools, molecular modeling tools. Is it? It is right. We have been studying about the drug design, molecular modeling, in design. About. Is it possible or not now? Yes, we have been doing it, but no. Just imagine combine design part, AML, the more number of variables can be brought in when you bring number of variables. Okay, this particular fragment is going to offer this kind of property. This particular side chain is going to produce such kind of safety. Based on the large amount of the data reported so far with the different experimental drugs, experimental compounds, chemicals, as well as the existing drugs. So it's possible, right? Now, all assets, if you can incorporate into your design, saying that no, I wanted a molecule with result, I think it will give you a optimized molecule. Now you don't need 20,000 molecules. Probably you'll be needing a much lesser than even maybe 100 to 200 molecules are the only ones required for you to start that synthesis. So how many times the workload is decreased? How much you can save the time? how many people required now. So that's why AML can be there. But screening this not it's not going to the AML is not going to the screening, but actually what is happening is in silico screening. If you have the molecule, or if somebody give you 100 molecules, saying that the medicine is given 100 molecules and for the biological evaluation, first thing what we can do is I don't want to start our experiments. If I start the experiment, just one MTT say to study anti-cancer or not, just second. Sorry. That screening, see, just imagine one day takes 3 to 4 days minimum, and 1 second. So for you to do so better at the protocol, how much time is required? But if you have MCL tools, screening to couple with AML, what can be what is happening? You know that no, this particular feature or this particular thing, this particular group is going to hydrophobes, it's going to DNA, it's going to the sensitization, it's going to adverse effect. You can screen them that no, you don't need to do all the molecules. You can actually reduce the number of actually filter out molecules. Look, this molecule is showing so and so pattern, and this pattern, because of this pattern, is going to be toxic. It is going to produce adverse effects and do that, right? So touch screen is possible. Drug repurposing definitely possible because drug repurposing is nothing but drugs you are going to use for the some other application. Based on the proteomics data, genomics data, what are the data we have so far generated with the particular drug? You know that no, this particular drug when taken for some condition is also altering another particular target. It is also altering the another pathway. You know that. Now, but the same pathway is found to be having a change in such other disease condition. Now, what particular drug which is already in the used for anti-diabetic active regime, also having activity in fibrosis. Example, metformin. Metformin is anti-diabetic. It was used for insulin sensitizer, but now later it was identified it's a AMPK activator. AMPK activator. Based on the AMPK activation, it's been proposed for the fibrosis, lung fibrosis. There is a paper came in Nature Medicine. More than 100 publications are there. So how it is it possible? Though they used AML tools at the time, but now metformin has been experimented well. But if we can use AML tools, we have more than I think 7,000 existing drugs. We can use these drugs property, you can able to identify which target is inhibited by altered by which drug. And this particular target, this particular pathway is found to be having a role in particular so and so disease, including common disease as well as orphan diseases, rare diseases, right? It's possible to have repurposing. Clinical testing definitely can have a role because how much you can do that's where number of patients, number of parents to evaluate which patient all a human can do the error, but not the mission, right? That's why you just have to do the program that no this many data sets, this many parameters. I think it can actually how you're going to select the patient because each patient might have done 100 tests to prove whether all the functions are proper. In the health patients, how many parameters are now? So that's how you select the right patients by using these tools as well as the evaluation after the giving the drug also possible. Natural products also can natural product drug discovery can also be possible. It is possible. Uh, so far using different products, but we are not sure and what are its mind and what is up we have been doing is we just on and pharmacology model saying that no yes drug is effective. But how it is effective, whether that's really going and built in the particular or not, particular receptor or not, we are still not demonstrating. That's why our natural product research is fragmented. So until unless prove that no mechanism of action, the drug will take it. This is a drawback with an that's where you can also again. So that means can help the drug development by analyzing amounts of the data more efficiently and accurately traditional methods. So traditional methods are highly time consuming and also chance for to be because we optimize many things. Whereas of course modeling and silico tools have used a little bit But now if we can couple this molecular modeling and uh, computation method, actually going to the improve for example, it's been used. Predict the efficacy of the compounds, predict the toxicity of the, and it's very, very important, right? More efficacy, compounds safety evaluation takes more time. Animals is required for you to do the pharmacology model is only 100. Maybe animals are required. Just 200 animals to 500 animals are required to prove compound is active or not. Whereas for you to prove the compound is safe or not, you need to prove each system, a single exposure, multiple exposure, 2 years exposure, all this to be done. That's why see a number of studies required are much higher, and number of animals required will be approximately 20 animals are required. 1,000 animals are required for you to prove whether the compound is toxic or safe. That's why when you're trying to optimize the design by silico tools as AML, you will be focusing more on the physical things where the solubility and viability and protein binding and all. You're also spend a lot of automation and the safety aspects are right. That's and also it can uncover genetic and molecular mechanism of diseases. Now, you know, the informatics you have it, right? What is bioinformatics? It is study, different pathways are the different significant pathways are connected to each, particular path system. Now, that itself is very huge. If they see any mixed data, more than 10,000 proteins, more than 5,000 proteins these are altered. How it is humanly possible if somebody give you one data set and ask you to do analysis? How are you going to do? You cannot able to apply a tool here. So what is required in the AML is you need a data set. You set data set, go and analyze the same protein, how it is normal, how it is the disease control. It's going to bring a pattern. That pattern also, what type of pathway? G protein coupled receptor pathway? Is it a receptor pathway? Whatever the receptor pathway it is, or what kind of enzyme it is altered, it can be grouping it and tell you that no, okay, in this particular rate is this things are altered, these particular proteins are altered. Based on what I can do is I will uh, try to normalize that particular protein which is found to be having a overexpression. Right. So like can use it. Same thing what we about put it here and target identification can use the ML, how you use the identification ML tools because humans have more than 20,000 2,700 drug targets. How many targets? 2,700 targets. How many targets we have explored? Probably 100, maybe 200. There are so many targets which we are not properly explored. That means we are not done yet. But scope for us to keep on target and which target to be taken decided by the target identification, which can be done AML tools. Don't ask me how that can be done, but this you have to invest more time on. But it will, right? So not always couple receptor. It cannot be always tyrosine kinase receptor. It can be something else. It can be ion channel. It can be enzyme. It can be a protein. You need to understand each target and each protein. We have the crystal structure of the protein or not because if you have the crystal structure of the protein or the target, how do you study whether your ion is binding or not, the inhibit is binding or not? You will not able to study. That's why that's possible. The compound screening and discovery, we talked about clinical development, we talked about small molecule optimization, we talked about clinical developments where you can actually um, we can predict whether compound is going to be toxic or not by using tools. Of course, you have toxic studies. Of course, you have animal studies. Number of studies, number of maybe doing it, you are not probably not going to fail. That's why it will be quick. That means when you have to apply the molecular modeling, or if you have to apply the AML and the molecular modeling, molecular docking and molecular dynamic simulations investigate the SAP protein and system using a binding frequency and geometrical properties. That means your target protein is having a crystal is going to interact like how it's bound. Let's see the first import PDB in data bank, drug bank, and PubChem. So PubChem are actually Uniport and PDB are actually structures. What are you trying to say? By using this tool, we are going to take those 2,700 targetable drug targets we have, we are going to they take access based on this. We have the drugs available. From we can how to select the target protein, and this is the let's assume this is a protein. How it three-dimensionally or crystally it is actually organized. Now, other side you can have the drug bank. You can also have the pubchem. These are some of the few of the many existing the chemical libraries where chemical chemical structures have been deposited. That can be a approved drug, that can be a molecule, that can be a simple chemical. So what you do is, you pick all those compounds, those chemical moieties from that thing. You can call them ligand. Try to dock it now. How particular protein is binding to your ligand, which is having a proper binding energy, free energy. We want a less binding free energy to bind to that. So that means so easily it's going and fitting into your target. The better the hydrogen bonds, better fitting it is. Probably it is binding to the target. That probably that it can be a potential candidate for you for the thing. Now if you have bound with the library, you think that some of the structures are binding it. That is some idea. Okay, the molecule is having this particular property. Now it can be fitting into it. Now, what next you can do? What next you will be doing is, because existing you will not be able to chemical library. You're going to have a no developed based on the initial hint that you're finding. Now you keep on changing the chemical structures, moieties. Optimize it. One variety which is a little bit binding can be optimized by using a in silico tools again. The Innova design, you can actually have different sides, how it's giving a score. What score you have? We are giving a score of binding free energy. So that means one molecule which is just having a weak interaction through your protein bank and library. By using that, you are going to design 100 molecules, thousands of molecules without synthesizing it, again designing it. Now you screen all the molecules again in docking, how they're binding now. So we are assuming that, because keep in mind of what are the properties required as per the Lipinski rules, what are the properties required for the toxicology rules. You probably will design a molecule which is much better than the molecule what you have identified. That's how it is done. Artificial intelligence based discovery based drug discovery is right than the new drugs. This is little a complex flowchart. So people who work in the artificial intelligence and machine learning, we people are lagging here. We don't have the basics in this. People talk about the artificial neural networks. People talk the decision tree algorithm. People talk about instance-based algorithm, whatnot. Support vector mission, key nearest neighbor, convolutional network, neural network. So these some of the words you might have heard about it. Really have tried to understand more. At least I didn't try to know about it because what are the reason. But the people who want to go into artificial intelligence, these are the some of the tools. You need to have idea. Once you know the ABCD of it, once you know the language of this, easy for you to integrate the drug discovery knowledge into. It's very much possible. Right. That's why we all have to, the faculty who are actually working there, if we can spend time on this particular basics, I think the way you do the research, the way it is are going to really change. So this is just a figure when I just put it here, but frankly speaking, I am also not aware of all these languages. But whenever you talk about artificial intelligence, machine learning, these terminologies have to come. That's why I put this slide here. So again, the conventional drug development process have a target, all the step by step, but every stage you can use your AML. So like now uh, starting from uh, designing the molecules to how we are going to filter them in sequence. This all can be done uh, uh, through your AML. So again, uh, that means at every stage it is possible to integrate. Now, while using AML tools, techniques in drug discovery, again, also there is a category like supervised learning, unsupervised learning, regression analysis, classification, clustering, and dimension reduction. These are some of the tasks they do, and there's a method mentioned in the and also repeated application. And again, these tools we are actually layman in this particular the tools. Maybe some of you might be having some background of it. If these are the things now happening in the drug discovery space, okay, people are trying to use so and so tool to get particular application done, and reference also given. So the point here is it's not just one tool. Yeah, you need multiple things like software. You'll have different programs and different tools for each activity. Here also we have now. What we have this? Hello, sorry, is somebody trying to talk? Okay. Now, so we are talking about the tools. We are also talking about where from you get the data, right? Live compounds and activity databases. Now, you know, right, where from you get the data. If you don't know the data, where from you're going to screen the molecules. That's why reference is the PubChem, Bell, Bank, Zinc data bank, binding D, ADM. These are some of the data banks where they're going to either talk about the receptor, the protein interaction, pharmacochemically, they have been. That means whenever you're thinking of using a tools, you need to have these data banks available. That you can screen them. After screening it, you know that now any hit there, any molecule is trying to bind to your target or not. Bind and promise. That means this particular is compound by now list of target and chemical data. Why from you will get the target title. If you don't know the structure, let's say one you don't know the chemical structure of the crystal structure of the protein, how are you to design a molecule? You know, right? Which is a binding pocket, what kind of amino acid are orientation it is there, which are the groups we are going to bind to your target. So you should know which are the molecules going to participate in the interaction with the ligand. That's why you need to have the databases for target also, like TBD, protein data bank, Uniport, Atom 3D, TTDs, like in the therapeutic target databases, molecular nets. So these are some of the data bank we have given the so websites also, so that no, you just have to go to that and find out whether the target is covered in that or not. Once you have the target, then you can import, you can download particular protein structure into the tool or the tool you're using it. Now import the data set, previous chemical bank. Now you try to dock it, whether they are binding or they are reacting or not, whether they are target or not. You do it. List a list of AI-based softwares for discovery development analysis. So AlphaFold 2, I'm sure you might have had a in a Google tool. Deep Bin Bind, Deep Deep Bar, Deep Cleaning, Deep Chain, Deep and Deep Purpose, Deep Tox. Deep Tox is nothing but the toxicology. AutomNet, Path, DSP. So what are these tools? These are the AI tools which are actually used in the drug discovery development. So why can't we explore all this? Some of them must be freely available. That's why you should spend now for us to use these tools. What basics required? We need to know the knowing ability to test. You can test, right? It's in English. Similarly, knowing that those AML, the common network tools, what you call it, networks and all those tools, we're not able to use these particular things. It's need to to now add another mention to our learning now is nothing but ML. I'm sure this really required for us to ensure our probably we are going to be highly effective to the many people in fields basically because we have a very good background of the pharmacy, but we don't have a good background of this now. If we combine this, it's a wonders. And so far, the people working in this particular ML in the pharmacy are not very few of them are pharmacy background. Most of them I interact with many people. Just trust me, people with the chemistry background, no pharmacy background, just chemistry background. People with engineering, people with electronic background are ready to do this by using AI. Advantage for this is we understand everything about farm, but no, not there is this particular course. Or if we can interact, if we make a team with those people, that what is that the purpose subject to possibility. If you don't learn at least team with the people, most of your college must be having also engineering. Engineering is compulsory subject. Now you have a multiple meetings in first round. First not have a lot of leads happening. Let it be a very huge meeting, but one trigger point may give you some hint to further. Then nobody will stop you. Right? That's what I'm trying to make here. So again, we already talked about AML can reduce time, cost, and improve critical testing. Lead discovery can be done efficient as all possible. This tool, again, I talked about talks, deep game. I'll talk about some of the tools. Now, how the startups are coming up in the AML by using AML in the discovery. A lot of startups come not just for the pharma fields as well. In the pharma field also, lot of companies are coming. Example, so these are things. Regional product proportion, like which other regions are controlling it. Ambitious organizations, like category wise, they have given pharma corporations, lead tech corporate. If you see it in all the sector, in all the segments of the pharma, USA, the first by USA, for European, then followed UK, then Canada. Majority of the again inventions through countries. Where are we? We the crores of the lakhs of people living background, lakhs of music. This is the really time to think, right? And just we know it. One Apple organization can able to control so much of the economy of the world because of the innovation and patents and all that's because ES is not knowledge driven or innovation driven economy. They produce any small all what is they invent it, pay it, and give the right to somebody else. They enjoy the luxury, right? The iPhone we are using today, the iPhone we are purchasing, we are giving it to USA because inventors are US people. That's how it is. Now, the drugs, if you don't discover here, the costs are going to be very high. Or is maybe manufacturing in India, they have to pay the amount royalty to the company for next 10, 20 years. So, either drug is not going to be available to us, or we have to use the drug, that's going to be very expensive. The reason is because we are not inventors. Now, if we have the tools like AML, at least if we can patent it with you, you can dictate, you can one other molecule which we have proof of concept is there will be people wanted to take your molecule. For that, very intelligent design of the molecule, optimizing all the aspects required, not activity. More than that, have more than 100, 200,000 different steps needs to be optimized. And it's possible you need to do any hard work. But will do it. What required is your intelligence to molecule. You should understand what is that you want to have it. Then get it synthesized. It's possible to synthesize in a laboratory and just do studies, animal studies, in vitro or in vivo studies with in collaboration with wherever you do it. And that no, my compound is active. Yes, it's active. I can be reproducible. This is one time study. It whenever I repeat it, it's going to happen. It and probably it's safe also. I have done so studies. Why people will not take that? Once you are confident my molecule is good, there will be so many people wanted to take that molecule. So since we cannot further develop it, it needs more, invest more people and more infrastructure. You're not able to do it, but no, you are the inventor of the molecule. Remember. That is possible. That's possible. By sitting in the academics, it's possible. Now, industries are also trying to do like this. Just trying to do the work like that and trying to outline to somebody. If you have one done like that, you're going to have very good success. What I'm saying is, India is still not in a see. There's no mention of the India. It's only mentioned Asia. Asia in Asia. China is said. Then also mentioned the Asia, right? So some of the success stories, like compounds by using AI tools, compound already gone to phase two trial. So when the compound gone to phase two trial, means they're actually progressing properly, right? Phase two trial have a efficacy study, a small population, and later phase more the large set of population. So at least even going to phase one is a good thing, right? I am we are talking about two, we already have molecule gone to phase two trials, right? Maybe it's not a very latest 2023, but I'm sure there must have been more movements happened. This is old the table. So none of the companies are well-known pharma companies. We are talking about the Pfizer, we are talking about the Merck, we are talking about the this LV. Of them are actually pharma companies. This is a small pharma organization, including Silicon Medicine. The company name is Silicon Medicine. This is the first one to repurpose that drug by using a clinical tools. So that means compounds. There are success stories across hepatic dermatitis, diseases, inflammatory diseases, COVID, as well as the diabetic and the cancer. Now, if there is a lot of collaboration happening, right? I'm sure this is very, very right. But what I want you to go through the next, what you should know, there are just a second. Okay, somebody is trying to call. Okay, so thing is there are left side Roche is there, Pfizer is there, nobody is there. Sam and Takeda, Eli Lilly, not AstraZeneca, all are this very giants in pharma sector. These are the leaders in the pharma. What are trying to do? Collaborations. Collaborations with what? AI organizations. Why they are trying to collaborate with AI organizations? Because they know that the future of the drug discovery, the future of development, drug development is going to happen through your AI. That's why trying to collaborate with the startups or no, yeah. If not the startup, established area organization. Why this is required? Because if you don't couple that, they still try to do the only the discovery of the temperature molecules 50 years of time, they lose the running race. And there's going to be more failures. They could afford, but not today because of this AI. So collaborations are the key. Are you also, if you want to collaborate it in you have to do it. If you have a good tool, if you have a good lead, there are people to collaborate with you. This you can actually go and communicate, contact with them, say that look, I have new target is validated. I have designed the molecule, and some of the initial data I have it. Do you want to collaborate? What kind of understanding we can have it? What kind of share I can have it? What kind of royalty I can expect? These are possible uh, currently. I may be working it, but it's you can think of. Just working the patient is not going to take anywhere. I'm telling this after publishing more 200 papers with in the institute. So I'm saying this because just should not be a publication required for us to along we can think beyond that. That's what I want to. So I hope what we talked about so far is this. We are talking about how the AI is going to impact. It's a broadly, I have not taken an specific example because we just started and in fact, after 10.5 years my time in Hyper Hyderabad, just as a faculty here, I realized I have done a lot of natural products work, just published more than 160 papers from here and a lot of grants. Now I realize if you don't think of discovery, innovation, we are not going anywhere. you're not leading anywhere, you will not benefit. The faculty, you need to have passion to think up new drug discovery. And benefit of that is, I think no salary you have to pay anybody, right? It's just no academic institute. Since we are government action, our student get the fellowship, we will get the service. We have all the setup, very good setup, literally very good setup. Do the if we are not up doing it, do this. That's where I have been insisting on my patients wherever the possibility. I'm asking them attend the AML workshops at the conference, and also now collaborating with some of the experts in this particular field, because my strength is I'm a pharmacologist. I can do a lot of animal studies. I can do a lot of in vitro studies. But if I don't two molecule coming, what kind of pharmacology I can do? And I have to depend on somebody. But if they're not using AML to like any system, right? That's why I need have or I need to collaborate with the people who are actually in that particular field, design the model as per my and give me a compound I can do, right. That's why I'm trying to do this thing. So that is half of my talk is on the overall cover of the artificial intelligence and machine learning role in the discovery and development. If I have, I think I have half an hour time, right? I want to talk about my research where we have done some work and where we have the ML tools. Murli garu,
Yes, sir.
hello.
Yes, sir.
So another I have.
Yes, sir. Sure, sir.
Right, okay. So yeah, for we talk about broadly. So some questions, you can just keep it with you. I think we can discuss it. Now, I have been trying to use since I mentioned of the drug development. It can come from the novel products. It can come from the natural products. It can come from the biologists. You know how important biology. But for you to produce a biologic, you need to have a difference. Conventionally, we pharmacology people are very good at small molecules. Now boom of biologics. So let's keep the biologics at the timing, because there is huge growth happening there. But no, not every pharma institute, every pharma organization had to start this biologics right away. So it's like monoclonal antibodies and that's different. But about the novel targets, we can also target the natural products. We have taken compound nimbolide. Nimbolide, we all know it. Um, Probably this is a compound gave me a lot of recognition for doing a research. When I started in 2014, I was trying to use traditional products like ashwagandha and neem. When investigation research, we found that nimbolide from the neem with a fernier from the ashwagandha are the most active moieties in these respective plants. So based on we have procured them from the suitable, and we try to evaluate in different animal models, because as I mentioned, my strength is pharmacology. So rather repurposing it, I was trying natural products. Use nimbolide. I've done a lot of fibrosis studies in cancer. We also done studies in models. So what we did with nimbolide is, now see, this study we have done in 2019. What we have done, we all know nimbolide in a condition, TNF-alpha, tumor necrosis factor alpha, are increased because it's inflammatory cytokine. And the inflammatory cytokines are increased to aggravate the inflammatory condition. That's where we have seen a different organs getting failed. Mostly in COVID, lung failure was happening because inflammation. One of the very important mediator of the particular inflammation is the TNF-alpha. Studies we are doing before COVID itself, right? So what was ending to the TNF-alpha, so it's binding, it's interacting, it's that's inhibiting it. That we have proven it through the in vitro protein expression by the confocal microscopy. We can see the middle is the in the which is in volatility. And also bio model of acute respiratory distress, that's also called SARS, severe acute respiratory distress, which that's what COVID, when COVID person affected COVID, you can see such kind of severe inflammation in the lung. Why? Because the inflammation is nothing severe immune cell infiltration into the tissue. The tissue you are talking about here is alveolus will have lot of cells so that no fluid is also accumulated. It's edema formation. Due to edema formation, there's no oxygen excess in place. That's the lung failure happening. So when we did this first study, we got a brown color, is an intense color in the tissues is nothing but TNF alpha expression increase in the disease model. So how did we use the disease model? We use the lipopolysaccharide, bacterial endotoxin, lipopolysaccharide. If you inject this lipopolysaccharide directly into the lung, it was causing the inflammation. Now, when we in the nimbolide, you see that particular the TNF alpha expression was decreasing. That means we have proven that nimbolide is inhibiting the alpha at the molecular modeling level, at the cellular level, as a tissue level. That it is actually having an anti-inflammatory activity. Based on that, we did the acute distress syndrome model, and we found that this compound is protecting the lung cells, lung epithelial cells, as well as the RA cell, macrophages from the inflammation. Expression studies, some of the studies, for you to prove whether they are not, either you have the protein level, you have to see whether particular protein expression is or not. How do you do it? You cannot do the biochemical assay. Either it can be Western blot, it can be ELISA, it can be some microscope. So what we did was we done the different set of because we have the tools to do it, and we demonstrated that nimbolide is protecting the inflammation by altering the some of the oxidative stress and inflammatory pathway. So as I mentioned, how do you induce lung function? If you want to induce any acute lung injury or chronic lung injury, lung disease, what are the inducing agent you want? You can send that compound directly into the lung. How do you do it? It's called oropharyngeal administration. That is, the compound like lipopolysaccharide for the acute lung injury, mycin for the chronic lung injury, fibrosis. You can open the mouth and pull the tongue out downward and contents, because of esophagus all the contents will be directly go back here. So that's one technique called the oropharyngeal administration. We are showing a lung image where, because section of the injury, you can see the lung weight altered as well as the lung morphology also altered. And we also histopathologically is that when you give the lipopolysaccharide to the animals, this is a lung sip. The animal control is nothing but the lung normal alveoli. Yeah, large cases. This is the to the body, no, so the bacteria in the lung. All the immune stand there and trigger the inflammatory response. That means inflammatory lung. Treatment with a different concomitant, you can see the improvement at the level. Also prove that through non-functional paralysis. So published in a called cell death and very tough journal published, publishing groups. You see the date, just see it's the date 2019. The COVID has 2020. So this has a very relevance to because all the mediators are having a very important role, very determined role in induced inflammatory lung edema, pneumonia, you say lung failure, right? Basically, this was happening. So we have done this particular study. We have known about the COVID. I've been working on injury models. We could demonstrate that. Of course, we cannot give this the patients. The reason was doing pharmacology is much easy, but doing your toxicology needs more time, more compound, more investment. We could not do that, right? Similarly, we also done that. We have moved to another model. Okay, in the fibrosis, we are, and not acute lung injury. We have seen it. What's happened to psoriasis? Psoriasis is also inflammation, stimuli hyperpollution of the keratinocyte. Basically, control is having a outer epidermis is very small, whereas the epidermis is thickened. That's also occurs. Acidosis is nothing but increased layers because of the hypertension of the keratinocyte. This is the actually the type. When given the Imbolide as a subcutaneous application as well as the topical gel, we could see your thickness of the altered. Thickness means it's the relation proven at the immunohistochemistry level. Imicamide, the chemical used to induce the process. Imicamide and use imicamide, where you see the this 67 is a preparation marker. TN4 is a marker. ICAM is a molecule stimulating the cell to accumulate in these expressions to be altered, decreased in the nimbolide treatment. That means in two models, able to prove that no nimbolide inflammation as well as reducing the inflammation of the character. All like any other natural products, nimbolide is also having a own disadvantages. It is not orally available. This is one of the biggest drawbacks that you could not able to make this basically. Right? Why you need like a curcumin, which is also able to be a compound as a critical that are in the poor availability category. Similarly, here the nimbolide is poor available. We have done the study. So that's why we could not take oral. If you take oral, also we don't know it's going to the blood. So how do you know? Can actually broadly that all the natural products are all available. You cannot say that. We have to do experimentally. That's in the pharmacokinetic studies. LCM liquid mass spectroscopy. In the pharmacokinetic study, you give the compound only as well as IV. You collect the dose at different time and estimate the concentration in the plasma and see when I that amount of the drug, how much Cmax concentration plasma, then you pharmacokinetic data analysis and say that no, this is the Cmax, this is the AUC, this is the relative oral bioavailability by comparing the. That's what we have done. So this LMS peaks because we quantify the drug in the is based on the mass because the mass is very unique characteristics of the each molecule. So we based on the mass, we quantify how much is the drug is reached to the blood. We'll be doing it right. The first time. If you see this figure, why is its concentration nanogram per ml? And none of the doses 10, 30, and 50 mg, none of the doses are crossed more than 35 mg per nanogram per ml. Nano per ml. 35 nanogram per ml. Only nanogram amount it can go. Just now, if you give IV, only 5 mg per kg. See, the highest concentration was more than 5,000 nanogram per ml. That means, yeah, obviously, if you inject IV, you're going to have a high concentration. That's expected. But in oral, only 35 nanogram per ml. That's the problem. Oral is not oral available. Saying it, percentage over less than at different doses. That means take 100 mg, only 3 mg. 100% goes are not 3 mg, less than 3 mg only reaches to the remaining 97% not absorbed. Is a problem. C and C also we could not make them as a oral drug. Another thing, do you think Nimbolid is available? You know how much we paid? We have paid 1 lakh rupees for 1 gram of Nimbolid. Itself is. You should ask me a question, sir. Why are you using such a high chemical? See, it's a sentiment, Indian sentiment, right? It's our plan. We all know the name is our property, and somebody get a patent on this, you all upset. Look at a patent, but stop you not doing it because you cannot do the natural product as such patent. What is required? You need to make a formulation. You need to make an analog of it. Because product cannot be patented if you understand the patent rules. You need to have the formulation. You need to improve the existing thing. For you to improve it, you should understand what is the problem with that, right? That's right. Second, just second. హలో హలో నేను ఒక టాక్లో ఉన్నాను. మీకు కాల్ చేయను బ్యాక్. యా సారీ, ఐ గాట్ ఎ కాల్ ఫ్రమ్ అడ్మినిస్ట్రేషన్. So you need to understand. So here I'm saying is, yes, it's active. If you only inject parenterally, that, but can you make the away? The cost is effective, it's so high. That's why you need to understand the problem. Then you have a problem solved through that can actually and problem right. So now, what are the ways to improve the pharmacological activity, pharmacokinetic properties? Either you can formulation, you can also make a analogs. You can also make analogs. So collaboration can. This person can isolate the nimbolite quantities. A chemistry person can make it. Like, for you to make analogs, you need to keep in mind the lipid rules. Okay. Since molecule is not available, I want to add such moiety, which is going to improve the water solubility. It can actually improve the solubility. Like that, this now once the molecules since chemistry lab, then that can go to pharma. Or if you don't make the analogs, okay, since polarity is not taken, I want to make a formulation. A formulation can be a nano formulation, it can be whatever you want. You can add sublingual also. People are doing it. Right. Right. What I'm saying is, we have done this particular study, we have made a lifelong formulation. We have proved no, it's much better upon inhalation administration compound in the same AIS model, and again published that work in nanomedicine. I'm not saying that here, but you can find my publication. We also done that study. So trust me, we published more than 15 to 20 people just with bolide. That tells that if we actually have a tools, the good thing for me is we have a good support with chemical tools. Imagine some other institutes don't have a proper setup. That's why you have to pick the right area for you. For me, it was limitation. For some limitation. That's why you have to pick the right area. Don't just seeing my work. Don't try to plan to do the work. Just see the feasibility. So that's very important for us, right? So since I'm talking about the lung disease models, now little from the nimbolide. So natural parts we talked about. Now we are talking new targets. In the targets, we all know that we already discussed here that due to COVID, immediate effect is lung inflammation, mainly in the lung, followed by kidney, liver, and other organs as well. Why these organs are failing? One of the reasons is severe inflammation. What are the mediator inflammation? We all know prostaglandins, we all know mediators. But one of the concept is there is a process called the netosis. Netosis. What is netosis? Neutrophil extracellular traps. Neutrophil extracellular traps. So these are this process playing an important role in meta. Why? Wherever there is inflammation, neutrophils are regulated. The neutrophils, upon action by these infections or the stimuli, they release some part of the DNA, out of the. So that DNA contains certain moieties, myeloperoxidase, neutral elastase, pepsins, reactoxins, and matrix cellular proteases. Why this particular some part of DNA is coming out with the other moieties is because this is antibacterial, parasite, infective basically, right? It's going to kill either virus or bacteria. It's good. It's defense. What excessive that happening in the conditions like COVID or any other condition where your lung is filled with a lot of neutrophils. These neutrophils are the nets. Nets are nothing but extracellular traps, which are part nuclei along the other the biochemicals are released out, and that start triggering inflammatory response. Inflammation response, that uncontrolled inflammatory stimulus is causing your lung is able to cope up. That's why I'm saying a lung. Now, how are you going to get this process? What is the enzyme responsible for this particular process? We target it. Where we are trying to explore, right? Okay, just a second. Trying to something. Yeah. Uh, NETosis targeting approaches. There are two ways you can target it. Somebody read in the pharmacology. One of the drugs for the cystic fibrosis treatment is Dornase, also palmozine, also called DNase 1. DNase, but A is going to be Dornase 1 or the Dor alpha or the palmozine. These are the names given. Why this particular drug is using cystic fibrosis? Cystic fibrosis with lot of sputum excretion, secretion, and the sputum is also very due to the sputum thickness, it's going to block your respiratory. Now, in order to reduce the thickness of the sputum, they are using a enzyme called DNase, which is going to break the extracellular DNA because of the DNA viscosity increase because the DNA in the fluid is going to increase viscosity. The moment you take the DNA is known as alpha, it breaks the extra DNA, so sputum is loose, so that no blockage will be prevented. This is the base for using that FDA approved drug for cystic fibrosis. The same concept can be applied here. The deal is the same DNase which is used as in the cystic fibrosis can also be injected in the muscle because nets increase, nets are increased, circulating through the response. If I target that, if I can know by using a DNA enzyme, that's one approach. Or you can also target the enzyme which is responsible for production of this. The enzyme called PAM, because peptide arginine 4, type 4. Type 4 is actually a rate limiting enzyme for the netosis. That means either I will prevent the formation of the netosis to add for DNase, which is to neutralize the already produced netosis. How it can be done. Now, if you see this figure, not only the COVID, in different conditions like sepsis, arthritis, rheumatoid, like systemic erythrosis, and ulcerative colitis, and fibrosis, and all the different conditions, nets are having a role. Not only there's a lot of literature saying that these are also playing a role in the metastasis. So there means not just one drug this can be targeted. If we can have a proper inhibition, there are the disease can be controlled. Now, so if you see this process, of the histones, the stones start at the which these condense nuclear events release the net. Now, what we did was we are making a smart nano ways to target the netosis is you can degrade the netos, or we can also inhibit the pad for enzyme. Right? What we did was we have taken an article, and area is a serum oxide nanoparticles. Serum oxide nanoparticles, if you just search in the PubMed, there are thousands of articles coming. It's a very good nanoparticle, nothing but just serum oxide itself is a trace. It's a very good antioxidant and anti-inflammatory. I have done a lot of studies on this for the last 10 years. So we know that it's anti-inflammatory and antioxidant. Now, using is not sufficient. But we try to code that nanoparticle, might be called DNase, which is why because it's not stable injection, right? If you can put in the nanoparticle surface, the stability is increased. So I'm making a smart nanoparticle, which is going to produce an inflammatory and oxidant property through nanoseria. I'm going to coat with this one DNase, which is going to be the available nets in the body. So we made this particular the smart nanoparticle system and tried it. One approach. Another approach, what we did, we tried to design the pad for intelligence. This is so two different approaches, right? Now, this is good as an idea is good, but you need to have this right. Right away going to a model and do this. That's why you right model to scale. So networks is happening not you can able to study in the differentiated leukemia cells, cell 60 cells. So we have used cell 60 cells and stimulated them poor 13 days state, 13 days state PMA. We call it. The green color in increase are then some kind of structures are formed, thread-like structures are formed. Thread-like structures, nothing but your nets. Control left side, no structures, intense green, which is indicated are formed. So this in vitro microscopic evaluation, the nets are formed. Because if you don't particular tool, how will you able to see the molecules? Now we have it. Image video, I could not able to display the video, but no, I can see we have done that time lapse imaging, right? You do in few minutes after few hours. This process taking place, you can image from starting to internet formation done it. We had that no nets are video can be taken by microscope. Later, we proved these molecules either the smart nanoparticles or the pad for inhibitors in a separate model or the pulmonary particles. Long complication of acute inflammation is the lung disease, right? Now, what we did, I mentioned nanoparticles which nanosyria coated with DNAs. We had done the lung functional parameters by the whole body photography, breathing see tidal volume, minute volume, or compromised LPS induced control model, whereas our not nanoparticles have really improved the performance. And these are the lung physical images are saying that, and also we have proven, oh yeah, the small what we have developed was this is better dexamethasone, which is used standard inflammatory immunosuppressant uh, used in certain conditions, right? So that what I'm saying, what found was, it's basically. It's a nanoparticle. Of course, we're only presenting a small piece of the data, but we have data. Don't want to show here. It's not just one specific study, right? But we are going to discuss. It's where Syria plus enzyme is come. We wanted it. That's why we are not published the work even. Work is done back, but we are not published. The reason is go for patent. Publicity cannot patent. That's why we're waiting for this. And the other side, we also done the pad for studies because we wanted to come up with a new pad for inhibitors. Because if you can remember, pad 4 is a rate limiting for the netosis formation. So we wanted to do the pad 4 studies. We in silico in collaboration with the chemistry department here and in biocell and now, before going to the initial studies, as I mentioned, it had the protein data bank. For each protein, you have a different protein structures. You have all the protein data bank and try to dock it. You also have to take existing inhibitors reported by different companies. You see, BMS, BSK, Jupiter Biosciences. These all companies that started working on this, and they have molecules. They have different stages of the clinical development. But what I'm saying is, this particular target is really having a good potential. That multiple companies are trying to work in this particular inhibit the PAD4 inhibitors. So before going to actual design, you need to understand what is the current status, how this particular inhibitors or the PAD4 inhibitors are interacting with your protein of different types. We have the comparison by using a Schrodinger software and how they're bringing to your PAD4 protein with this. You see, this molecule which is binding to the interacting is by the docking PSK molecule as well as the standard inhibitor and also different the scores we try to understood it based on also have the molecule designed by our chemistry department also try to do this particular set could not get good activity. The fall we have another molecules and the documents we had done the value in the ingredients we had done the uh, screening and if you see it molecule a very good activity better standard inhibitor chloramidine. So this C molecule, we found that this can be further. The binding to this molecule simulation molecular dynamic simulation we have done it, and then we realized that no, what are the molecules we have designed is actually having a good interaction. So based on this, we have understood the document. This molecule to our pad for it. Then followed, we did the in vitro studies in the cell-based studies. Green color representation of the net estimation created by few all separated compounds, and most six is having a better activity. That means the what compound we taken for further is worth taking. This fourth one. We have done this. We also done the citrulline H3 because citrullation is taking this. The section also found decrease microscopically in experiments proving it here. The molecule that selected 36 is a better active one. Then followed by we did in the in vitro in vivo model of the acute respiratory distress syndrome. Disease LPS in model. There is a trace in the this parameters upon treatment no molecule. These are the lung represented images. How the lung actually look and up with the new molecule, oral as well as internally. This is a histopath to that no showing a activity. Then there is we have mentioned a lung fibrosis and the lung complication, acute inflammation, the severe inflammation. We have in the lung function, the breathing frequency, minor to volume, and resistance, NH, and these are all the inflammatory points which are having a role in this are bound to be altered upon treatment. E molecule is nothing but a new for inhibitor, right? The histopath and then magnesium staining is extensive blue accumulation. We start magnesium staining very we don't see any alloys here, right? That's what the lung fibers, and we could see a pigment is reduced, inhibited the collagen accumulation. That means the new pad for inhibitor chemistry is still having an activity. We also proven through the chemistry. You have good studies, then more can be become a good patent application. The collagen application, and for these are the decrease treatment of molecule when compared to the existing anti-fibrosis than PF is like 500 at this, and we have model we have studied acute pancreatitis. What we are trying to do, inflammation happening in depth. We have acute respiratory syndrome. Now we are curious whether it is going to affect in other model or not. We done the acute pancreatitis. Cerulean induced pancreatitis model, LRG model. The seen a protective effect of molecule. Why we have to do it? Because more experiments you do, more confidence you have that no producible that my compound is protective. The doing more is one is you, this is done, you'll have that application. You also have confidence that no this particular compound then is effective. Done the study and we have seen product effects. I last week, if we have patients continue, we have targets. So what about is we have natural products with the smart nano. We talk bad for inhibitors. Now we have another target. It's a SUT3 activator. SUTins are actually enzyme, very important enzymes which maintain a radar balance in the cell. What we identified was no SUT3 levels are in the fibrosis and also in the inflammatory conditions. We try to develop the new SAT3 activators. So that's what our hypothesis was, because there are no standard activators are available. Only natural products are there. We all know that natural products have a problem. That's why to design SAT3 new novel actors to protect against acute respiratory syndrome as well as pulmonary fibrosis. This was our hypothesis. Known activator of the SAT3 suitable. Surprisingly, though it's a known activators, we have invited only one or two are there. Something is wrong. Compound is cannot be feasible as studies. They're the first one to do the studies in the expert diseases and lung fibrosis. There is antioxidant in 2D line models, and also in the cell in the lung epithelial, where red color, which is indicated that in the dark color, is nothing but it's happening with the uh, a toxicant like bleomycin, and we are seeing a cell death upon treating the C12, which is not molecule, which is a already non-molecule. Also done the study of pulmonary fibrosis. So we are doing. We want to validate target activators effective or not. So we understand. Okay, this compound activation are affecting the are effective to lung from the acute lung, chronic lung disease, but pulmonary in this disease. Based on that, we have designed molecule. So the solubility because these molecules are not designed based on the molecules. This is the problem. If we don't work together, if we don't understand the importance of applying the tools at the design part inside the molecule, the activity level, it is not in my control. A molecule behave the way it's supposed to behave. No, it is in your hand, right? That's why at the design part, you need to make right more design. This unfortunately molecules are having, but of this, G22 is the one which having better uh, activity with the ICP2L the 4.4 molar based on the docking poses. Then we had an in vitro style contraction study, but how the it's a gel is a assay the fibrosis level. Gel contraction means there is a fibrotic state. The gel which is having a cells will be contracting. Contraction is the fibrotic response. So this is receive contract able that's why we done still as a as a questions is west to my 22, which is safety is important than anything else. That's why you have to keep in mind of all some of the important aspects of while while actually designing the molecule designing stage itself. That was really missing in some of some of the experimental evidence I have given it. But otherwise, it's a very good field where we can explore it, and if we can work together, if we can work in teams and with the other experts, there is a possibility to for us to progress it, right? So I'm satisfied with what we have done it because it's producing a thesis, it's also producing the publications and patent. But no, until unless you don't commercialize it, there's no value. But no, it's not a one-day job, or it's a not 1 year job, right? It's a process. We are in the process, right? Now, it is my story I'm talking about, but for me, because based on my experience, what can be done from your side? Institute level, what could be, no need to take this in a just giving a little hint what we can take it. You can take a simple model, simple, very easy to do develop models. We don't take a very complex model because the model itself is going to take a lot of your energy, a lot of your funds. You need to have a very simple model. which is easy to develop it, and also select the compound which are simple to synthesize it. The criteria easy to develop and evaluate the condition. Some of the disease. So why I'm telling is one of the limitation for many people is the model. If you have good model, your formulation scientist can design the molecule, your chemistry person can design the molecule, your pharmacognosy person can design the molecules. All can analysis person can also do the pharmacokinetics. So that's why I thought you can pick some of the disease models like psoriasis, allergic contact dermatitis, burns and wound healing, arthritis and inflammation, lung disease like ARDS and lung fibrosis, renal fibrosis. There is surgery involved, urinary urethral obstruction, renal damage like cyclosporin, cisplatin induced one, amputation B induced one, hepatic model like paracetamol and carbon tetrachloride induced lung injury models. I mean, these are some of the examples I have given because I have worked in most of the models. Tomorrow you can pick the model. What is required is you need to read the articles, mostly focus on the model, how they are developing it. And you need to right away see, simulate that are you going to, are you in a position to develop that model in your lab or not. If you are not able to develop in the model, you depending on somebody else to the activity is not going to work out for you.
Sir, your screen is not visible. Sir, Chandraiah, sir. Hello. Sir, Chandraiah, sir.
Hello?
Yes, sir.
Okay.
Now it
Did,
is
did,
okay.
you, did, you, did you miss any of my last few minutes?
Just last 1 minute, sir. And
No,
we
I
can't
was
see your
saying is,
screen also.
thank you so much for the patience, and also thank you for the opportunity. I hope I have conveyed, something which is going to be useful to all the faculty to take it further. That's what I'm trying to say.
Okay, sir. Thank you. So is your presentation is done?
Yes, I'm done.
Yes, sir. Thank you. Actually, because
Okay.
last 1 minute or 2 minutes, we missed your presentation. That's why.
I know. I just closed it. And okay. So
Okay.
could you able to see my this institute level models? You could able to see that?
No, sir. Just last 2 minutes we had the disturbance. That's all.
You want me to go through that? I can go. Or if you're okay,
Yes, sir.
we can stop.
Yes, sir. Please, for 1 minute.
Okay, let me show that also because window. Press again. Just a second. I'm going to share this again. Okay, what I'm saying is this. This slide, did they witness? Did you see the slide? Hello?
Yeah. No, sir. Now it is.
So what I was conveying was, see, those we wanted to do some discovery work. So these are some of the potential, simple animal models to develop it. Simple models. We don't need a lot of skill to do it, and then you can start evaluating your formulation of your chemistry based molecules, or your natural products which we might have developed it can be studied in this particular conditions. So because just thinking of very sophisticated hi-fi model maybe not feasible for us, for some other institutes, right? Maintenance is costly, the inducing agent may be costly, or surgery may be involved in that, you have to do instrumentation. So to avoid that, I think these are some of the models we can quickly take it up and see how they're working. Later, you can go more deeper. You need to have some proof of data, proof of concept data for you to further discuss with anybody to collaborate it. Without the data, nobody's going to take. That's why I'm saying it. And with this, I would like to thank the all the organizers, principal and vice principal of the college, Dr. Murli and Dr. Samyukta, and all the faculty members we have joined from different institutes. Thank you so much for the for the patient listening. And I would like to take the class, questions if you have any. Thank you. I'm just escaping from my presentation. Yeah.
Yes, sir. Thank you so much for your thorough presentation, sir. So, dear participants, now the session is open for Q&A. If you have any doubts, please ask your questions.
Many of them are leaving actually. It's like, okay, good. That is still there are 84 people. What was the highest number we could see? I could not see that.
134, sir.
Okay, good.
Okay, Dr. Chandraiah, can I ask a question?
Yes, sir. Yeah.
Yeah, just now in your presentation, you showed that you have worked on HDSC3 and you have shown translocation using microscopy.
Yes.
Can you just please elaborate, because we were looking for something similar, and is it related to HDSC3 activity, or is it just inhibition of translocation? How?
Okay. So I have to share the screen again, but let me tell you what we have done it. What we are saying is, if we inhibit the TNF alpha upstream, so it's going to prevent the translocation of HDAC into the nuclei, right?
Okay.
So we are preventing the translocation to the nuclei.
Okay.
We have that inhibitory kits also available, but we are not done the kits. We have done the translocation to the nuclei based on the fluorescent microscopy.
Okay, but can we correlate it with HDSC inhibitory activity, or it is TNF inhibitory activity? That
Obviously, if you inhibit the HDAC, what is happening is, it is not going to activation there itself is translocation, right? That's possible.
Okay.
Yeah.
Yeah. Thank you.
Yeah. You are from which institute, sir? Patilji?
I am RC Patel Institute, sir. We had contacted you for that UOO model. We had contacted you earlier. We talked with you earlier.
What model is that?
Unilateral ureter obstruction model.
We are using that model.
Yeah.
So you are successful using that model.
Yeah, we could do that.
Very good model, right?
Yeah, right. Yes.
That's why I mentioned this model for somebody to see. Renal condition is very severe condition. Chronic renal injury damage, end-stage renal damage. So many people are there in India mainly. So there can be a research done. Yeah.
It was because of your guidance that we could successfully do that.
Excellent. Wonderful. So happy to know that.
Thank you, sir.
Yeah. Anybody else?
Yes, sir. There is 2 questions in the chat box. I'll be reading out for you.
Can you read? Actually, I'm not able to. This font is slow.
So someone is asking to share the AI-based platform or softwares used in the studies.
So the one I have shown in the PPT, they wanted me to show. I can share that.
Yeah, yes, sir.
There are links given there. They can actually go there. Actually, links. There are links given there. So you can share. I will share those slides to you. Then they can actually try to go through the links and try to see whether they are downloaded or not.
Okay, sir. Sure. Thank you, sir.
Yeah.
And there is one more question. Can you suggest only any in vitro research areas for B-Farm projects?
Sir, in vitro, in the sense, you need to have cell culture facility. Cell culture itself is a very expensive thing, right? So that better to use, tissue-based experiments. Again, tissue-based experiments are banned now, right? You cannot use it. You have to go to slaughterhouse and get the tissues if you want to do the tissue-based, some experiments. Or maybe you can think of zebrafish models. I feel for B-Farm based level, these are some other things can be think of. Directly going to the rat model is not going to work out. Cell culture facility is tough for them to handle it, right? So.
Okay, sir. And one more question. They are asking for, are there any courses for artificial intelligence, machine learning for pharma people?
Okay. What they tell is first you learn the basics, like ABCDs. There is no exclusive program for pharma. They think is no for everybody the same language. Now you have to integrate it. Once you know the language, you have all the pharma knowledge. You can able to put it together. First, you should any conventional AML course you should take it. All those programs which required Python and data science needs to be learned first. And there are so many programs happening, right? If you have time, I think they should enroll. Trust me, this is really going to uh, dominate our pharma science field. Job potential can also significantly increase.
Yes,
You know,
sir.
yeah.
One more question, sir.
Yes.
He's asking to suggest an easy method for extraction of Nimbu in small scale.
Okay. So there is a procedure available. I didn't do the extraction because I'm pharmacologist. Though we collected the leaves to do it, but no, we could not do it. But I think we have to follow the literature basically. I'm sorry, I don't have the expertise in this, but we have procured. I mentioned I have procured from one of the supplier. So I don't have this expertise, but no, it's possible to extract it. But there's a lot of literature also available. Simple oxalate extraction or simple extractions are there. I also have to follow the literature only.
Yes, sir.
Yeah. I hope I didn't bore you guys.
No,
One
sir,
thing
not
and
at all.
Sir, good afternoon, sir. Sir,
sorry, madam.
this is Samyukta. Good
Yeah,
afternoon.
madam. Yeah.
Sir, thank you very much for your informative session, sir. It's very informative, which will cover vast topic, target validation, preclinical testing, clinical testing, everything, sir. Here, I have one question, sir. Can you predict any drug side effects or toxicity, sir, by using this AI?
Yes, you can predict it, right? You can
Yes.
predict it.
Uh,
Predict it as well as the design. You can design as well as you can predict it. Both you can do it. You can predict as well as the design also. First you design it.
yes, sir.
If it's already molecule is already available, you can predict it. Because you'll avoid those particular structures which are going to be problematic. That's what you are going to design level it as. But you already have molecule with you, whether that's toxic or not, you can predict it by using it. You have a software to do this, right?
Yes.
Yeah.
By using some different
Yes,
softwares, you
yes,
can.
yes. There are certain rules and structure based, rules, structures like rules for property. Also
Yes.
rules for the
Okay.
toxicity. Reactive groups, the particular heterocyclic ring. These all say existing from literature only you can do that. Software cannot predict it without having a data. So you need to have a set of data which is available. Based on that, it is going to
Discover and
predict it.
everything. When you are implementing whole data, we will get that
Yes,
appropriate.
exactly. Yeah.
Yes, sir. Thank you so much, sir. Thank you.
Yeah.
Yes, participants, if still there are any questions, you can ask.
Any more
Otherwise,
questions?
there are any questions, you can ask. Otherwise, okay. So someone is asking like,
Looks
what are
like
the
lunchtime. Looks like a lunchtime.
organizations where they can do AI/ML methods?
Is a question?
Yes, sir. What are the organizations where they can do AI and ML methods?
Organization means for us or for overall?
Overall.
Overall, specifically, I cannot able to tell this because many of the IITs are offering the certificate courses, right? Then they can get enrolled in that. But as an organization, we have a triple IT Hyderabad, which is there, right? Actually one of the first few to use this, including pharma plus in different other fields as well. So triple IT Hyderabad is could be one of the potential, and I'm sure all the other IITs also doing it, including IIT Madras.
Okay, sir. Thank you.
Yeah.
Okay, I think all the questions are done. So we are moving to the last part of today's session. Now I would like to invite Mrs. Srilata ma'am, Assistant Professor, Department of Pharmaceutical Analysis, for conveying vote of thanks to today's speaker, Chandraiah sir.
Okay.
Thank you, sir. Thank you, sir. Good morning to all. I, G. Srilata, Associate Professor, Department of Pharmaceutical Analysis, MLRIP, consider myself very honored and lucky to propose the vote of thanks for this magnificent EFTP. First of all, I would like to thank our respected speaker, Dr. Chandraiah Godugu, who managed to find time in his hectic schedule to attend this special event. And we are all pleased and delightful that the day 5 session of EFDP program on artificial intelligence in public health has been conducted extremely well. Today, we are very lucky to have eminent speaker, Dr. Chandraiah Godugu, Assistant Professor, Department of Pharmaceutical Sciences, Naipur, Hyderabad with us. Sir delivered a talk on opportunities in drug discovery and development in the era of AI machine learning. Sir, we thank you for such a wonderful presentation, your passion for the subject, and letting our participants feel interested to learn further about AI-involved drug discovery by emphasizing on various topics like target development, target validation, lead optimization, preclinical testing, and clinical trials. Sir, thank you for emphasizing about AI machine learning, their network, algorithms, and various widely used AI techniques, bioactivity, chemical databases in drug discovery, and also discussing about various AI-based softwares, AI tools for drug discovery, development, and analysis, and various examples of drugs that got discovered through AI, and also about AI ML based startups in drug discovery and collaborations of pharmaceutical organizations with various AI organizations, and also about Nimbolide, a natural constituent, and also on various other topics. And also, sir, thank you for discussion about the opportunities at the institute level. We thank you for enhancing our knowledge on importance of AI in drug discovery. And my heartfelt thanks to all the participants for joining the session and making it more interactive and interesting.
I'll appreciate
extend
this.
my sincere
All the
thanks.
participants.
Yes, we have to appreciate the participants. Yes, extend my sincere thanks to our management, our beloved chairman Mari Lakshman Reddy Garu, Secretary Miss Shreya Reddy, MLR IP Principal Dr. N. Jyoti, Vice Principal Dr. Arnav Malik for their unwavering backing for this initiative. I thank last but not the least our energetic organizing secretaries, Dr. Bala Murali Mohan and Mrs. Samyukta Mehta, who continuously kept their efforts by working round the clock to make this EFDP a grand success. Now, I would like to rest my words by once again thanking each and everyone for making this event a huge success, and hope we will meet soon with many such events. Thank you, each and everyone. Thank you all.
Yeah. Thank you all. And just to inform you that whenever I go to Genome Valley, I used to go for jail inspections. I used to see your college. When you go on the outer ring road, it's there, right? When you go on the outer ring road, that your college is
Yes,
seen.
Yes, yes.
So I used to see your institute,
Yes, sir. So thank you so much, Srilata ma'am, for conveying vote of thanks. So now it's time to wind up our 5-day faculty development program. As the organizing secretary of this EFDP, I'm honored to express our collective gratitude to everyone who is involved in making this faculty development program a resounding success. And I'm very much privileged to announce that this is our first ever faculty development program. We received more than 300 registrations from across the country from many renowned colleges and universities. This program success reflects our institution's commitment to faculty development. With this, we also have gained valuable knowledge, skills, as well as network from various colleges. First and foremost, I extend my sincere thanks to our distinguished speakers, Dr. Anant Naik Nagappa, Father Sabu Thomas, Dr. G. Shweta Kumari, Dr. Pankaj Kumar Singh, and Dr. Chandraiah Godugu. Your insights into the integration of artificial intelligence in the pharmacy sector, nanotechnology, drug discovery have been incredibly valuable. Your expertise has truly enriched our understanding of these critical topics. So here just I'm going to give a gist of all the 5 days of our faculty development program. So on the first day, we had Dr. Ananta Naik Nagappa sir, Director, Om College of Pharmacy from Karnataka. He has discussed the impact of artificial intelligence in the pharmacy sector, employment opportunities, personalized medicine, medication management, technological changes, challenges in healthcare, and the importance of teamwork and the role of artificial intelligence in patient care. On the second day, we had Father Sabu Thomas, Associate Professor from Kochi, engage the audience with a discussion on different artificial intelligence tools offered by elaborating on applications like ChatGPT, Gamma AI, Google Gemini, and Zero GPT, and how these can be utilized in community pharmacy. On the third day, we had Dr. G. Shweta Kumari, who is the assistant professor in the Apollo Institute of Medical Sciences and Research, Hyderabad. She had elaborated on how artificial intelligence is transforming various fields, including pharmacology and pharmaceuticals. She provided insights on drug discovery, development, machine learning, deep learning applications, clinical trials, and pharmacovigilance. On the fourth day, Dr. Pankaj Kumar Singh, Assistant Professor from Naipur, Hyderabad, spoke about nanotechnology, its challenges, impact of artificial intelligence, cancer diagnosis, targeted delivery, development of nanocarriers, and their role in cancer treatment. Then on the final day, that is today, we had Dr. Chandraiah Godugu, Assistant Professor from Naipur, Hyderabad again, discussed opportunities in drug discovery and development in the era of artificial intelligence, which includes target validation, lead optimization, preclinical testing, clinical trials, NDA approval, bioactivity database, and more beyond that. So for all these things, we appreciate all the respected speakers for your time and effort you took to engage with us, answering participants' questions and sparking meaningful discussions. Very, very thank you for that. And I would like to thank all those faculty members who have joined, registered for this EFDP program from various colleges and who have attended for this program. You are also one of the main people for the success of this program. Then I want to thank our management, Sri Mari Lakshman Reddy Garu, Sri Mari Rashekar Garu, Ms. Shreya Reddy Garu, who always being a back support for us in conducting the events like this. And a special thanks to Dr. Naka Jyoti, Principal MLRIP, and Dr. Arunabha Malik, Vice Principal MLRIP, for encouraging us to organize this event. And a big thanks to my fellow organizing secretary, Mrs. Samyukta, Assistant Professor MLRIP, for this wonderful idea of organizing EFDP and her constant support in every task we came across in this journey. And last but not least, I thank personally each and every faculty member of MLRIP who have contributed their support in their individual level and joined with us in making this program a huge success. Thank you one and all. Thank you so much.
Thank you.
So with this, we are going to wind up completely our 5-day faculty development program. So you will be receiving a final feedback link to all the email IDs. So those participants who have registered, you will be receiving feedback links in your email IDs. Please fill the feedback form. After filling the feedback form within a few hours, you will be receiving your e-certificate also. Thank you so much. Thank you once again. Thank you, Chandraiah sir. Thank you so much for your time. We'll meet you soon, sir, if possible. We'll meet you again offline for some other event.